ওকে, তো আইসোলেশন এন্ড ক্যারেক্টারাইজেশন অফ ব্যাকটেরিয়ার মধ্যে ফার্স্ট অফ অল যেটা আছে সেটা হচ্ছে যে আইসোলেশন ক্যারেক্টারিস্টিক ব্যাকটেরিয়া। তো এখানে কিছু ব্যাকটেরিয়া এখানে ফিগার দেওয়া আছে। দেন এটা হচ্ছে একটা ব্যাকটেরিয়া ফিগার। তো এটা হচ্ছে ব্যাসিলিয়াস সাবটিলিস, দেন হচ্ছে কি পোর্টিউয়াস ভুলগারিস, এবং তার সাথে হচ্ছে কি স্টেফাইলোকোকাস এওরিস। তো each distinct circular colony should represent an individual bacterial cell or group that has divided repeatedly, being kept in one place, and the resulting cells have accumulated to form a visible path, and most bacterial colonies appear white, cream, or yellow in color and fairly circular in shape. Then definition of the bacterial isolation এর মধ্যে যেটা আছে, bacterial isolation is defined as the technique of separating one species of bacteria from the bacteria's mixed culture by different plating method like curing, separating, striking, and serial dilution. If we consider about bacteria culture, here we can see different kind of bacteria species here, but among these we have to separate one individual species, and this is called isolate. And this is, I think, the word was most used in the time of COVID. Isolation was the word was very much a famous word at that moment. So next, this process will be number one, pouring process, spreading process, striking process, and serial dilution process. Then next is the growth of bacteria can be observed over the solid nutrient medium. Growth can be observed where we can observe this growth, just in the solid nutrient medium, then liquid growth medium, then some automated liquid culture medium, then some automated liquid culture medium. So these are the things where you can observe this thing. Then next, to visualize and isolate the bacteria in solid media, we need to add bacterial suspension into or on the media. Okay. So to visualize and isolate the bacteria in solid media, we need to add bacterial suspension or on the media, into or on the media. So this is the thing. Then oppositely, the bacterial inoculum in the liquid broth is characterized by turgid media. Oppositely, bacterial inoculum in the liquid broth is characterized by the turgid media. এটা characterize কিভাবে হয়ে থাকে, এটা হচ্ছে turgid media দিয়ে এটা হচ্ছে characterize হয়ে থাকে. Then automated liquid culture media also detects the bacteria present through various characteristics like productions of the carbon dioxide. Then therefore the bacterial isolation provides an important tool to study the morphological, physiochemical, and pathogenic properties of the bacteria that has been isolated. Okay, so therefore bacterial isolation provides an important tool to study the morphological, physiological, and pathogenic properties of bacteria that has been isolated. Then isolation and characterizations of bacteria, the shape, size, arrangement of the isolates and their differentiation into gram-negative or gram-positive bacteria were found, and the bacterial isolations were characterized biologically, biochemically by various tests like indole, MR, methyl red, and BP, Vogas, Proscure, and citrate, etc., including carbohydrate fermentation. So the bacterial isolations were characterized biochemically by various tests like indole, MR, BP, citrate, etc., including carbohydrate fermentation. So this is in the identification and characterizations of bacteria. Then methods of isolation and methods of inoculation and isolations of the pure culture. Methods of inoculation and isolations of the pure culture. Then mixed bacterial population, sputum, urine, pus, then infected wound, then abscess, etc. This is in the mixed bacterial population. Pure culture is single kind of microorganism growing alone in protected environments. We can say the pure culture, bacterial colony, mass composed of identical bacterial cell. This is the bacterial colony. Then methods for the isolations of the bacteria. What is the method? Then curing method, then spreading method, striking method, serial dilution. First of all, pure plate method. So first we can see here the pipette bacterial sample onto the petri dish, then pour liquid nutrient agar onto it, then squill to mix, then incubate, then colonies grow on the agar surface and the subsurface, and split plate method. And first of all, here we can see the uh, pipette bacterial sample onto the surface of the agar plate, then spread sample evenly over surface and incubate it, and colonies grows onto the surface. So this is the spread plate method. And uh, one what this is called, uh, one spreading tool is used to spread this thing. And in this case, we could not see any uh, spreading tools here. Only squeezing to mix is happen here. Then pure plate method versus spread plate method. So pure plate method here is 1 or 0.1 ml, spread plate method 0.1 ml only. Then here the incubation, empty plate, and bacterial dilution. Here we can see. Then add melted nutrient agar onto it. Then swirl this mix, and then the colonies grows in or onto the medium. And next is the inoculation plate containing solid medium. Inoculation plate containing solid medium, then spread the inoculum over the surface evenly. Then spread the inoculation over the surface evenly, and colonies can grow only on the surface of the medium. And striking of the bacteria on the nutrient medium. Then mixed culture of bacteria, here you can see. Then different striking pattern, we can see in the next figures. Striking method, first of all we can divide this plate into 4 quadrant. First of all, this is the first quadrant, second quadrant, third and fourth. You can see. Then strike and quadrant strike plate method, you can see here. Then here you can see, after striking, the colonies has started to grow here. Then A, B, C, D are four plate. Uh, Here is the first quadrant, second, third, fourth. Now we can see here striking done here. Then area 1 is heavy confluence growth, you can see here. Then area 2 is the less dense growth. Area 3 is the weak growth. Area 4 is the single growth colonies, we can see. Then sample to be counted, the samples we want to count, we have to dilute it. Then how we can dilute it? Then if we take 1 gram of the sample and make it 10, this is the 10 to the power 1, and 10 to the power minus 1. Then if we take from 1 ml from this and make it 10 ml, then it is the 1 by 100. This is the 10 to the power minus 2. By this we can make it like 10 to the power minus 6. So this is the serial dilution. And here you can see too many colonies to be counted. And here we can say 159 colonies, 17 colonies, 2 colonies. Here no colonies formed in this picture. So if we consider here, the colony number is 159, then the plate count number 159 into dilution factor, here is for number 3 is 10 to the minus 3. So here is dilution factor. So dilution factor and plate count. If we multiple these two things, then we can find 1.59 into 10 to the power 5 number of cells, which is the colony forming units per milliliter of original sample. Per milliliter of the original sample, then identification of bacteria depends on one microscopic appearance under the compound light microscope, which include reactions with the Gram stain and morphology and arrangement, capsulated or not, then motile or not, spore forming or not. This is the microscopic appearance. Then macroscopic appearance, then culture appearance, then biochemical test, then gene test we can see here. Macroscopic appearance, in my macroscopic appearance, culture appearance, biochemical test, then gene test we can see here. Then microscopy and morphology, shape. In shape we can see the spherical, rod shape, comma shaped or spiral shape. We can see here this shape we can see here. Then size is 0.2 to 1.5 micrometer in diameter, about 3 to 5 micrometer in length. Microscopy or morphology, arrangement, pairs, treaters, groups, chains, Chinese letter pattern. Here you can see. Then mortality, weight, hanging drop, and preparation. Then spores is shape and position. Here you can see different positions here. Then capsules, this is the capsules figure. Then culture media. Common integrations of the culture media is peptone, meat extract, NaCl and agar. Peptone, meat extracts, NaCl, agar and water. So uses of culture media to obtain the pure culture, identifying and recognizing of bacteria, and some media used to study the bacteria to be in bacteriological tests. Then type of media, according to this physical status, there is the liquid media, solid media and semi-solid media. Liquid media, solid media, semi-solid media we can see here. This is the physical status and type of media we can see here. Type of media we can see. Then use of the media. Then number one is the simple basal media. It uses for the cultivation of the common microorganism, but not for the uh, fattidious bacteria. An example is nutrient broth and nutrient agar. It uses for the cultivations of the common microorganism, but not for the fastidious bacteria. And example is nutrient broth and nutrient agar. Nutrient broth and nutrient agar, and then special purpose media. In this media, then blood agar, chocolate agar. It is simple media enriched with one of the following substances: blood serum and glucose, and it is used to cultivate the fastidious microorganisms, and just like uh, Neisseria, gonorrhea, and then Haemophilus influenza. This is the one of the in this media we can see. They cultivated fastidious microorganisms. So this is the fastidious microorganisms. We can see the Haemophilus influenza, then Neisseria gonorrhea. This is the thing. Then selective media. Selective media favors the growth of certain bacteria and inhibit the growth of other bacteria because it contains the inhibitory substance. Then uh, mannitol sugar agar, mannitol salt agar, to contain high concentration of the NaCl that most bacteria cannot grow in this concentration except the Staphylococcus spp. So this is the mannitol salt agar, contain high concentration of the NaCl that most bacteria cannot grow in this concentration. So this is the mannitol salt. Then this is the figure you can see. Then Maconis agar contain bill salt. Maconis agar যেটা আছে সেটা normally contain করে থাকে কি bill salt contain করে থাকে that inhibit all kinds of bacteria except enterobacteriaceae. Then Salmonella Shigella agar SS is selective for Salmonella and Shigella. Salmonella Shigella agar SS is selective for Salmonella and Shigella. and this is the Shigella flexneri and colorless colonies, and the E. coli with slight pinkish colonies we can see here. Then the Salmonella Shigella SS agar, and bismuth sulfate agar is selective for Salmonella PP. Then bismuth sulfide agar BSA, Salmonella SP, then uh, uh, Klebsiella pneumonia, then Pseudomonas aeruginosa. This is the three we can see here, 1, 2, and 3. Then ABC we have denoted here. Then bismuth sulfide agar is selected for Salmonella spp. Then differential media, it is used to recognize certain spp of bacteria, either by certain characteristic colonies such as fermentation or non-fermentation of lactose on Maconis agar, and this media contain neutral red as indicator which change in color during the fermentation because the gas will be produced and decrease the pH. Then inoculated, then this is the left, no lactose fermentation we can see here, right is the lactose fermentation with the certain effect of the media, hemolytic or non-hemolytic on blood agar, beta hemolytic, beta complete hemolysis, then streptococcus uh, pyogenes, Streptococcus pyogenes alpha hemolytic, alpha incomplete hemolysis or the partial hemolysis, then Streptococcus viridans, then gamma hemolytic gamma, no hemolysis, then Enterococcus fascialis, Enterococcus fascialis we can see here. Then this is the figure we can see for different bacteria that we have see before. So this is the core bacteria. Then microscopic characteristics of this microorganisms in any culture is, first of all, size. It can be very small, pinhead less than equal to 1 millimeter, and medium size 1 to 2 millimeter, large size is greater than 2 millimeter. Shape is circular, irregular, then radiated, rhizoid, filamentous. Elevation is flat, raised, low, convex, convex dam, papillate. This can be elevated. Surface structure smooth, rough, granular, ringed papillate. This is the surface structure. Is interlobate, crenate, then dentate, then carrelate, then filament. This is the ace. Then color and opacity, transparent, translucent, then opaque, then fluorescent, metallic scene. This is the color and opaque. Then consistency, but butyrus, then viscid and friable, membranous. This is the consistency. It is the emulsifiability, fiability, so easy or difficult, homogeneous, granular, membranous. This can be present. So first of all, size, shape, elevation, surface structure, age, color and opacity, consistency, emulsifiability, then phenomenon, swarming, Porteus, then Medusa head, Bacillus. Then swarming or Porteus mirabilis, and then Medusa head of the Bacillus anthracis. Then pigment, 10 pigment, exopigment. This pigment produce outside bacterial cell in the media, and Pseudomonas aeruginosa produce phyocyanin green pigment, and Pseudomonas fluorescens produce fluorescence blue pigment we can see here. Then Pseudomonas aeruginosa and Pseudomonas fluorescens, Pseudomonas fluorescens, then endopigments. This pigment produced inside bacterial cell, Staphylococcus aureus golden colonies, then Staphylococcus citrus yellow colonies, Staphylococcus albus white colonies. Mm, then odor. Odor is sweet odor apple like such as the Pseudomonas, bad odor fish like then the Proteus. Then 12 is the growth in the semi-solid media, gelatin test. And then 13 hemolysis in blood. Hemolysis in blood, and this is the reference.